you, Atira, for that very nice introduction. And thank you to you and Shriya and all of the other organizers of the Brain Awareness Week for putting this event together and for asking me to speak. So, what I'm going to do today is to tell you a bit about what I do in my lab. And before I start, this is my lab. This has most of the people who are there currently, some people who were there, you must be around. They're the people who do the work, so I'd like to thank them at the beginning. Okay, so what my lab is generally interested in is understanding how neural activity in the brain generates this vast range of complex natural behaviors. And by complex natural behaviors, what I'm talking about are some of these behaviors. And one of the axes, and these are both human and animal behaviors, and one of the axes that we classify these behaviors along is this axis along which, depending on what triggers this behavior. For instance, you can imagine that behaviors are triggered either by internal or external stimuli. And there's a continuum. Each of these behaviors could be triggered either by internal or external. To give you an example and to explain further what I mean, I'm going to start with this behavior right at the corner, which is sort of at the one extreme of this continuum that I'm referring to. This behavior is a knee-jerk reflex. So if you've been to a doctor's clinic, the doctor is likely to tap under your knee with a small hammer, and what happens is your knee jerks forward. This behavior is a simple reflex that the doctor uses to test nerve function in some sense. This behavior or this movement is driven entirely by an external stimulus. Occurs only when the doctor taps below the knee. And we've understood a lot of the circuit or the mechanisms by which this behavior is produced. This tap below the knee activates some neuron, and then that activates some other nerve cells in the spinal cord, and then that ultimately acts on the muscles, the extensor and the flexor muscles here, and pushes the leg forward. This is a behavior that's triggered entirely by external. Some other behaviors here, like for instance, cheetah catching the gazelle, is triggered by both external and internal stimulus. This the cheetah has to see external stimulus, a gazelle, for this behavior to be produced, the prey catching behavior. But at the same time, this prey catching behavior will be produced if the cheetah is hungry. If it is not hungry, then it's unlikely that this behavior is produced. So that's what I mean by it's triggered both by the presence of an external stimulus, the gazelle in this case, and the internal state of the animal, whether it's hungry or not. All of these behaviors here are can be triggered both by a combination of internal and external stimulus. We think that many of these behaviors on top are largely driven by internal stimulus. In my lab, what we like to understand is how are these behaviors initiated. Before I get to that, I'd like to highlight three important similarities between these different behaviors. One is that they all involve the coordination of many muscles. Two, they're very rapid behaviors. Approximately 10 seconds is the entire duration of most of these behaviors. And they can all be triggered very rapidly. For instance, this bear catching the salmon, the behavior, the bear catching the salmon is triggered soon after the bear sees the salmon. If that doesn't happen fast enough, then the bear is not going to get its lunch. Now, in addition to these, so what we're essentially interested in the lab is to understand how the brain converts a stimulus, be it external or internal, into the appropriate. What we'd like to understand are what are the neural events that lead up either from an external stimulus or a spontaneous self-triggered movement. Why is this important? Well, it's an important fundamental question in neuroscience, but we don't have a complete answer. It's also important for better understanding what goes wrong in diseases with movement initiation problems like Parkinson's. It's also important for restoring movements in paralyzed people. You must have read or heard about experiments where you can put in chips into the brains of monkeys and connect these up to muscles and restore function in monkeys that have been paralyzed, or this has been done in a limited number of humans, that put in wires into their brain and record the activity of the nerve cells, and then connect these up to robotic arms potentially, and provide some amount of function back to these people who lost the sense of function. And in order to do this better, we need to understand what connects stimulus to the appropriate movement. What happens? What happens in the brain as the stimulus is converted into a movement? What are the neural events that lead up to a? So this is currently being studied a lot. It's studied mainly in monkeys and in rodents, and in both these animals, it's studied with simple tasks. For instance, the monkey has its finger on a point in the screen, and this is called the fixation cue. And then a target comes up. And what the monkey is expected to do is to move his finger from here to here. That is, from the fixation cue to the target. Okay? And so when this go cue comes, that is either when the fixation cue disappears or the target changes color, the monkey is expected to perform. There's similar movements that are similar tasks that rodents are trained on, and I had also done tasks like this during my PhD. But essentially, these tasks are very controlled, and they've allowed us to learn a lot about what happens in the brain before a movement is. And we've learned that there's a lot of activity in the brain just before the start of a movement. This activity is referred to as preparatory activity or motor planning. And we think that this activity is important and somehow sets the stage for the start of the appropriate movement. So today, what I'm going to argue is that while these tasks have told us a lot and have informed us about preparatory activity and motor planning, these tasks, there are a couple of limitations to these tasks. One is these are not very natural movements that the animal is making. They're very simple movements, and it's difficult using this task to study internally triggered movements. But most of these tasks involve a cue being provided to the animal, and the animal has to respond to the cue with a particular movement. Okay? So while these tasks have told us a lot, and it's really very nice research that has informed us about what happens before the start of movement, I think, to paraphrase Lewis Carroll from The Walrus and the Carpenter, I'd like to say that the time has come to start looking at more complex movement. That's what my lab does. We use the zebra finch, a songbird, as a model system for studying how movements are initiated. And this is the zebra finch, the male zebra finch, Australian songbird, and this is the female zebra finch. And what I'm going to show you in the next slide is a video that will demonstrate the behavior that we study. That is the song of the zebra. What you'll see in the next slide is a male zebra finch and a female zebra finch on the other side. What you'll see initially is this male zebra finch is sitting in the corner, and then he'll get up and go and carry out his beat. Okay, so you must have all seen that. Let me play it once again for you. So what you saw is that this bird gets up, it walks across to this side once it sees the female, and then it produces a song which sounds like cookie, coo, cookie, coo, cookie, coo, cookie. And the song itself has is also there's also a dance that's along with this song. The whole song and dance is part of the bird's courtship ritual that it produces when it sees the female bird. Let me play this once more for you. Here's the bird. This is actually when the lights are off. As soon as the lights are on, the bird notices the female. You saw that cookie, coo, cookie, coo, cookie, coo, cookie, coo, along with the dance of the bird. So this behavior is a very nice behavior. That's a natural behavior. It's a complex behavior, and we can study this. And the way we study this, it has been studied in the lab for a long time now. Because the way we look at song, because any behavior, we need to be able to visualize it, be able to quantify it, be able to study it better. So here's the way in which we look at the song of the zebra finch. So this is again the male zebra finch. We can put a microphone in the bird's cage and we can visualize the song using what's called a spectrogram. So this is a spectrogram with time on the x-axis and frequency on the y-axis. What we're essentially doing is asking what are the different frequency components present in the sounds at various different time points. Using these, we can then distinguish the different sounds that the bird is making. What you'll see in this spectrogram is there are colored portions and there are white portions. These colored portions especially represent syllables or sounds that the bird is making. And these silent portions are gaps between these sounds. What I've highlighted for you are the 4 syllables produced by one particular. These 4 syllables, the patterns are very different. And what that corresponds to is that the frequency components in different sounds are different. If this A sounds has a different set of frequencies at different times, while the B and the D are more similar in terms of their frequency throughout time. Okay. The important takeaway from this is that sounds are different. We can classify them or we can categorize them in this case as A, B, C, D. And if you record an adult zebra finch over and over again, the bird will always produce these 4 syllables. And it will also produce these 4 syllables in the same order, A, B, C, D,
Excuse
A, B,
me,
C.
sir. Sorry to interrupt. Your voice seemed to be fluctuating a little bit. Could you please hold your mic closer?
Yes, sorry. Is this
Yes.
better?
Yeah.
Okay, thank you. Okay, so this ABCD, adult zebra finch will always produce these 4 syllables in exactly the same order. Of course, one zebra finch will produce these 4 syllables, in this case, one particular finch, but another zebra finch will produce a different combination of syllables. Important point is that they produce each zebra finch will produce the same set of syllables in exactly the same order throughout its life, once it's learned it and once it's become an adult. So this is a very nice behavior that we can quantify. Like I said, this behavior is part of a courtship ritual, which is triggered by the presence of the female. This is the natural behavior. This has some ecological relevance. So it's important for the animal to learn this song properly. Also, the bird does produce song when he's alone. And we think of this in the field, we think of this as practice for the song that he produces to the female. So the song produced to the female is what we think of as performance, and the song produced when he's alone is what we think of as practice. So here's a complex movement, a natural movement that's also produced when the bird is alone. So self-initiated or self. So what I'd like, this is a model system that we look at in the lab to understand how movements are initiated. And what I'd like to convince you by the end of this talk is that this is in fact a very nice model system. We have learned a bit about how song is initiated. Okay. In addition to the behavior, like I said, there have been a lot of people who've done very nice work on the zebra finch, trying to understand how song is produced. And what they've done also is to identify areas in the songbird brain that are important for song production. For instance, I'm just going to highlight 2 particular pathways in the bird brain. One is motor pathway, which involves multiple different nuclei. So each of these circles here represents a group of neurons or a nucleus that's important for producing song. And the way these nuclei in the motor pathway produce song is by ultimately connecting up to motor neurons that the midbrain and brainstem. These motor neurons connect to the vocal organ of the bird, which is the syrinx, and the respiratory muscles. And by controlling these 2 muscles, the bird can produce a song. And these nuclei in the motor pathway are important throughout life for producing song. There's another set of nuclei called the anterior forebrain pathway, which is important for learning song. So it's important to the young bird for learning song. It's also important in adult birds anytime the bird needs to change his song a little in response to environment conditions. Okay, so we know that there's a very nice behavior that's readily quantifiable. The bird produces his song when he's alone, and we know a lot about the brain pathways for producing song. What do we know about how song is initiated? Well, we know that song is initiated with a variable number of these short syllables called introductory. So here are 3 bouts of song produced by the same bird. Like I told you, this ABCD that I've highlighted here is produced multiple times within each bout of song. And this ABCD lasts about 1/8 of a second. So about not 1/8 of a second, but about 800 milliseconds. That is about 8/10 of a second. So these three bouts of song, though they contain multiple repetitions of this song, this ABCD, they don't start with this song. Instead, the bird starts with these short sounds called introductory. And what you'll notice is that the bird repeats it. The number of times the bird repeats it varies each time the bird sings. Here the bird repeats it 5 times. Here the bird repeats it 4 times. Then within a song, within a bout of singing, bird may repeat two songs together without any introductory notes. Sometimes he includes an introductory note in between. Sometimes he includes two. We know that song begins with these variable introductory notes, but we really don't understand much more than that about how song is initiated. So essentially, the question we ask in the lab is, how is song initiated? What are the behavioral and neural events leading up to song initiation? And for today's talk, I'm going to focus on the alone or the internally triggered song. So our approach is to say that this is a starting of the movement. A is the starting of the movement. And what we want to do is keep stepping back in time and asking, what are the events that happen before the start of the movement? Like I said, these introductory notes are the things that happen before the movement. Today, I'll describe some of the work that we've done understanding how these introductory notes are produced. Then I'll also describe some work where we've gone back one more step in time and asked what happens even before the first introductory. Tools we use are behavioral analysis, use electrophysiological recordings, that is recordings of individual neurons in awake birds. And these are, we also write custom scripts in MATLAB for our analysis. Okay, so this is the outline of my talk. I've told you what I'm going to, what the general question is that we look at in the lab. I'm going to show you some behavioral data initially, and the behavioral data is focused on this portion, the introductory notes, trying to understand how do these introductory notes progress to song and what controls the number of introductory notes, what is the origin of. Then I'll go on to some neural data showing you again related to how introductory notes progress to song. Then, like I said, move one more step back in time and ask what happens if the first introductory note is produced. So here are multiple bouts of song produced by the same bird. And here is the one spectrogram that I showed you. I've only shown you the beginning of that bout of song. And all of the other spectrograms for the 39 other bouts in this figure, I've compressed them. So don't worry about the detail. They're all aligned on the start of the song. So here, this green rectangle indicates when the song started in each of these song bouts. What you'll notice is that, like I said, these bouts of song begin with these short syllables called introductory notes. And these introductory notes can be as little as, two introductory notes at the start of the bout, for instance, here, or as many as 4 introductory notes or 5 introductory notes here. What you'll also notice is that the time interval between these introductory notes is a little variable initially, and it becomes a little more stereotyped towards the end. So what we do for each bird is to measure what are the mean number of introductory notes that a bird produces before the start of the song, which essentially means we take all of these bouts of song, count the number of introductory notes in each of these bouts, and take an average of that. That's the mean number of introductory notes. Calculate the time between individual introductory notes. For instance, this time, the time interval between this and this, these time intervals are in the range of half a second to about 1/10 of a second. And we also look at how similar these introductory notes are to what we call the last introductory. This is the last introductory. And what you'll already see in this picture is that uh, these introductory notes get a little darker and more redder as the introductory notes progress towards the start of song. And this getting darker and redder corresponds actually to the introductory notes becoming louder. The bird starts with this meh, 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 and then increases its the volume of these introductory notes. These introductory notes come much faster, and then the bird starts his song. And we had quantified this. This is some work that I did as a postdoc with Alison Dope. And what I'd done was to look at these characteristics of introductory notes. So here, what I'm showing you is the interval between introductory. across many bouts and across many birds. Each of these circles is data from one bird. These squares represent means across all of the birds. The x-axis here shows the position of the interval between interacting. So this G minus 1 corresponds to the gap or the time interval between the last interaction and the start of song. G minus 2 corresponds to the interval between these two interactions, that is the last interaction and the second, and so on. What you'll see here is that the duration of this gap or the time of that gap reduces as the interacting notes get closer to the start of the song. And that's what you see here. It reduces. It also becomes much more stereotyped. Now, another thing that I'd shown and what I'd referred to in the previous slide, what you'll notice here in this one example is that these interacting notes are much softer here as compared, or softer and more yellower, whereas here it becomes much darker, redder here. And this basically corresponds to these interacting notes becoming louder as the bird approaches its song. And many other characteristics of these interacting notes change as it progresses, as it gets close. But I'm going to only refer to this loudness for the moment. What I can ask is, how similar are these interacting notes to the last one? In other words, maybe how loud or relative to the loudness of this interacting note, how loud are these? Are they very soft? And I can compare those. And what I've plotted here is a particular measure called the distance. You don't need to worry about what the metric measures, but essentially it's a way to tell how similar these interacting notes are to the last interacting. Greater the distance, the less similar they are. The smaller the distance, the more similar they are. What you'll see again here is that these interacting notes progressively become more and more similar to the last interacting. In other words, they sort of get louder, 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 and then reach a particular loudness, and the bird starts its song. So this is some work that I'd done as towards the end of my postdoc, and we hypothesized that these interactive notes may in fact reflect some kind of preparation in the bird's brain to get started, to get a song started. This is what Divya, a graduate student in my lab, decided to ask when I started my own lab at ICER. Divya, a graduate student, decided to ask, how do these interactive notes reach this last interactive note or ready state? We call this last interactive note state a ready state. Bird's brain is ready to start song. What Divya imagine were two specific hypotheses. One is that just like musicians calibrate their musical instruments or their voice by initially playing the instrument and then checking and then tuning it. Divya imagine that maybe the bird's brain also does this. That is, these introductory notes serve to tune the vocal organ of the bird, and the purpose of these introductory notes is for that, to tune the vocal organ so that it's ready to start song. So here's this hypothesis in a cartoon form where every introductory note is produced by this motor system, and the height of this eye here, or the eye refers to the introductory note. The height of the eye refers to some characteristic loudness. This, when the bird produces one introductory note, this provides some feedback to the brain. The brain then adjusts the volume of the next introductory note so that it's louder, and this then again provides feedback until the brain realizes that it has reached the correct loudness and it can start the song. This is some form of calibration of the vocal apparatus before the bird starts. Now, alternatively, Divya, imagine that this could just reflect internal preparatory process. Rather, the brain is getting ready to start song, but it doesn't need this feedback. There's some internal process that's going on in the brain, and feedback is not necessary. In order to test these two, what Divya did was to say that sensory feedback is important according to this hypothesis for reaching this ready state, whereas according to this internal neural preparatory processes hypothesis, the sensory feedback is not necessary. So then what Divya did was to look at songs after removal of sensory. Here, what I'm showing you is two forms of feedback that are important in the one is this auditory feedback, which is feedback from the voice of the bird back into the ear, and feedback from the vocal organ, that is the syringeal muscles. Remember, I told you that the song itself is produced by both the muscles of the vocal organ and the muscles of the respiratory. And that's why again, it's although we are talking about speech or song or a vocal signal, this is still produced by a combination of the activity of multiple different muscles in the body. That's why it's a good movement sequence. This syringeal organ also provides feedback to the brain. That is, it's called proprioceptive feedback. This feedback goes from the syringeal muscles back to the brain, and it provides some information about the state of the vocal. These two forms of feedback are what Divya decided to perturb and look at what happens when these two forms of feedback. Here's an example of a song of a bird. After cutting the nerve that provides feedback from the vocal organ to the. So here's the song as a spectrogram before the surgery. You can identify all of these syllables. Immediately after surgery, the song changes a bit. All of these sounds become very similar in their structure. That's why you'll notice that these patterns are all similar. But the durations of these notes remain different. They remain very similar to what they were earlier. This is because the nerve that they are cut, not just the nerve that provides feedback, but the nerve that controls the syringeal muscle. Both these nerves are right there together, and there's no way to cut one or the other separately. This location. Okay. The reason, so the now the brain doesn't control the syringeal muscles, which is why the structure of these sounds has changed, and it's all become very uniform. The duration of these sounds is controlled by the respiratory muscles, which we did not perturb, which is why the durations of these sounds are still exactly the same as before, and so we're still able to identify these sounds as what. So this is what happens when we cut the nerve that provides proprioceptive feedback. In addition, we can also, in a separate set of birds, what Divya looked at was what happens when she removes auditory. It turns out when you remove auditory feedback, the song of the bird does not change, and this is something that others have noted as well. This is the song of that bird before surgery, and this is the song immediately after. So what Divya then asked was, in both these categories of birds, what happens to these introductory notes that occur before the start of song? What she found is in all these three categories of birds, that is the sham operator or control birds, the birds with nerve cut and deafened birds, we don't see any change in the number of introductory notes that the bird produced pre and post surgery. Each of these circles here is data from one bird, and these lines connect data from the same bird. These bars represent the mean and variation across the group. What you see here is no change between pre and post surgery in any of these categories of. We have looked at a number of other characteristics of introductory notes, and she essentially found that immediately after surgery, none of these characteristics. What this suggests is that these introductory notes, we can rule out the hypothesis that there's some form of calibration. Other, these introductory notes seem to reflect internal neural practice. And to sort of support this idea further, what Divya found was that, like I showed you at the beginning, this is one songbird from a bird, and the bird produces multiple motifs right next to each other without any introductory notes in between. This says that introductory notes are not essential once the bird has started singing. But what we find is that almost all of the time, introductory notes are present at the beginning. What Divya did was to ask whether they are always there at the beginning of a bout. What she found is that there are a small fraction of birds, in our case 25% of the birds, have multiple different ways of starting their song. So here's song from the same bird. 88% of the time, the bird starts his song with these introductory notes represented by I here, these short sounds. Whereas 6% of the time, the bird starts with something else. These vocalizations are different from these introductory. They're part of the bird's non-song vocalizations. They're used for other communicative birds. But 6% of the time, this bird actually starts with these notes. What you'll notice here is the song essentially is the same, A, C, A, B, C. There's no difference in the song. It's just how the bird starts his song. Okay. So this again told us that the actual sound that the bird produces is not important, but rather There's something about repeating a particular sound that seems to be important for the bird to get started. However, Divya also found there were a very small fraction of birds that don't sing introductory notes at all. Here's a bird. Here's one spectrogram of a bird that doesn't sing any of those repeated sounds before the start of his song. And here's many more spectrograms put together. Each of these sounds is replaced by a gray bar, same duration, and they're all stacked up one on top of each other. They're aligned to the onset of song. Again, what you notice is this is a song, and here there's no sounds at all. There's no variability here, like what I showed you earlier, that variable repeats of interactive notes occur before the song, but here there's no such thing. But just starts with his song directly. This is only 1% of the birds. In our colony, we have about 2 or 3 birds in a colony of 300 birds. And I'll get back to why these birds are interesting and how it still validates what the way we think songs started, or the function of these interactive. Okay. Quickly to summarize, what I've told you is that these introductory notes that are just before song, they seem to get louder and they seem to come faster as the bird approaches his song. And the last introductory note we think is some kind of ready state, which tells us that the bird's brain is ready to start song. And what Divya showed is that these sensory feedback is not essential. So the bird is not calibrating its vocal organ with these introductory notes. Rather, these introductory notes reflect something going on in the brain as the bird brain prepares. So at the same time, what two other colleagues of mine, a graduate student Shika and an undergraduate Vishruta, noticed is that different birds have different introductory notes. Here are the introductory notes of 20 different birds that have been put together in this one single spectrum. And what you'll notice is the patterns of these introductory notes are very different. So each bird has its own introductory note. And what Shika and Vishruta also noticed is that the mean number of introductory notes a bird produces before the start of his song is different for different birds. We know that in the case of song, each bird produces its own song. This variation in song between individuals is a result of learning. So a bird that sings a particular song sings that because it learned it from its father. But another bird, which is part of a different nest, learns a different song from its father. So what Shika and Vishruta imagine or hypothesize that this variation in introductory notes that we see, both in the structure of or how these introductory notes sound and the number of these introductory notes for the start of song, could also be a result of learning. What they did first was to compare. So juvenile birds grow up with a father in our colony, and they learn their songs from the father. Once the juvenile is old enough, what Shika and Vishruta did was to take these juveniles, these adults out now, and ask how many introductory notes do they produce? What is the structure of these introductory notes? How does that interact? And what they did was to compare these with those of the father. Asked, are these introductory notes that this adult bird produces, are they very similar to what the father produced? And here's one plot from their data showing the mean introductory note for the father and for the pupil, the son. Each of these circles is one bird, and these squares represent, and the whiskers represent the mean and the variation seen within a nest. And what Shika and Vishruta observed is a very nice correlation between the number of introductory notes that a son produces and what his father produces. Now, this correlation by itself doesn't tell us that this is learned. It could be hereditary, it could be inherited from the father, or it could be learned. So, to distinguish these two, what Shika and Shika did was to separate juvenile birds very early in their life from the father, that they don't listen to the father's song, and then keep the juvenile bird with another male bird who sings a different number of introductory notes. We refer to this other male bird as a social tutor. And what we've shown here is the father and the tutor are differently colored because they produce different number of introductory. And what Shika asked was, when this juvenile grows up, does it produce these pink introductory notes or the green introductory? In other words, does it copy the introductory notes of the father, or does it copy the introductory notes of the tutor? If it copied the father, then we know that this is inherited from the father. If it copied the tutor, then we know that this is learned from the tutor. And here's an example of a bird's spectrogram. This is the father's song. This is that of the tutor. And here is a song of the pupil. What you see immediately is that the patterns here in this spectrogram for the pupil are more similar to the tutor than the father. This is true for the song, like everybody else has shown before us. And this is true for the introductory notes as well. Here, these numbers represent the mean number of introductory notes that the this is very similar to what the tutor produced, not the father. This is shown in data for many other birds, where we're comparing the mean number of introductory notes that a pupil produces with what the father produced or with what the tutor produced. What we see is a nice correlation between the mean number of introductory notes that the pupil produced and the tutor produced. We know that this bird, when it grows up, it produces the same number of introductory notes as the tutor. It's clearly learning the introductory notes from social. The normal nest case, it was learning it from its father. Then what Shika did was also to tutor a different set of birds without interacting. What she wanted to know is, will they learn songs without interacting? Because remember, I told you that there was a small set of birds that don't produce any interacting. What Shika did was to separate these birds from their father. They've never listened to their father. Instead, she would play back songs without interacting notes to a speaker. And when this adult bird grew up, she asked, does this bird produce interactive? And to our surprise, we found that this bird produces interactive. This is a father's song. This is the song that Shika played back to the speaker without any interactive notes. And here's the song of the pupil. And you'll see that the song is very similar. So birds do learn from such playbacks of song from the speaker. But this bird produces interactive notes. And it produces interactive notes that are different from what the father. It's not inherited, but in fact, the number of interactive notes produced by these what we call playback tutored birds, was approximately the same as those were normally reared, socially tutored, and birds that have never been tutored. All of them still produce interactive notes, suggesting an innate tendency to produce repeated syllables at the start of a song. So to quickly summarize, I've shown you that these interactive notes reach a consistent ready state. These may reflect some kind of preparatory process, an internal preparatory process. The mean number of interactive notes per song is learned, although there is an innate tendency to start song with interactive repeaters. Now I'll move on to the neural data. First, I'll show you some data about what happens during interactive. So to do this, like I said, we have to implant birds with electrodes. Here's a picture of a bird implanted with electrodes, and it's connected up to an amplifier. We do all this with appropriate ethical permissions from the Animal Ethics Committee. We take care of our birds to make sure this entire surgery is done under anesthesia, and we make sure that these birds are comfortable with this. So the area that we put recording from, or the electrodes are targeted towards, is this motor nucleus called HVC finch brain. This corresponds to motor areas in mammalian brains, and in fact, there is some similarity to areas that are involved in language production in humans. And this area that we're talking about, this HVC, so we implant birds with these electrodes that are targeted towards HVC. And what I'm going to show you is a recording of one of these neurons. What you'll see here on top is the electrode trace, that is what we're recording with the electrode. What you'll see here at the bottom is the song itself. See that? In addition, you listen to the electrical signals produced by that neuron. So neurons produce these electrical signals called action potentials, and each action potentials, when you're listening to it, you'll hear it as a pop, as a. You listen to each of those action potentials. In addition, what you'll hear also is the song. So initially you'll hear only the neural activity. Later what you'll hear is both the neural activity and the song itself. So let me play it. Okay, I think I'm not sure whether, yeah, I think the sound was pretty clear. One, you would have heard this, which actually corresponds to these big deflections or the spikes or action potentials or electrical signals that this neuron produces. And what you would have also heard is the song of the bird, that cookie coo, cookie coo, cookie coo. What you'll notice here is that this neuron, even when the bird is not singing, this neuron has some amount of activity, what we call as spontaneous activity. And little before the bird starts singing, this neuron stops its activity, and then it has a different kind of activity during song. In fact, if you had listened to it closely, it would have sounded like a prop, because it produces multiple action potentials very close in time to each other. Then once the bird stops singing, you'll notice that there's a small period where the neuron is not active here, and then it starts its spontaneous activity. To look more closely at this, here, if I blow up on each of these events, these big deflections of the voltage that I've recorded with the electrode, this looks like, the waveform looks like this. Each of this is one action potential. If you overlay many of these together, they all look exactly the same because these are all action potentials or spikes or electrical signals from the same nerve cell. This is about a thousandth of a second in duration, so millisecond or a thousandth of a second in duration. These are extracellular, that is, we're recording from outside the neuron, and we get the information about when these action potentials occur. That's the information that we're interested in. What we're essentially looking at is when the bird is producing a song, what do these at what time does this nerve cell produce action? And I'm going to focus on the introductory note part of this thing, because again, song has been studied by many others before us. What you see here is the song spectrogram, and what you see here is a voltage trace for one trial. What you see are action potentials produced only before this last introductory node. These are all the other introductory nodes that occur before it. And what you see here, as marked by the red arrow, is there are no action potentials before these other introductory. In fact, this is very robust, and to give you a little more detail, we can represent these as dots. Each of these dots in this figure represents an action potential produced by a neuron. And what I've done is, this is work that Divya is doing in the lab. What Divya has done is to color code these introductory nodes in the form of red, green, or blue, depending on their position. This is the first introductory node in red. These are the middle introductory nodes in green. This is the last introductory node in blue. Essentially, what he has plotted here are 5 different neurons from one bird, and the activity of those 5 neurons during the different introductory nodes. 0 is the time when the introductory node was produced. These dots refer to action. What you see is some cells like this one, are active for the first and middle introductory nodes. They're active only before the red and green introductory, not before the blue ones. Whereas other cells here are active only before the last introductory. This already has some indication of what is the position of this introductory node and how is what's going to occur next. So this already indicates what's going to happen next. So this is ongoing work, and we're still figuring out what happens as these introductory nodes progress to song. Okay. So what I've shown you now in the neural part of this is that the neural activity in this particular part of the bird's brain, a motor area of the brain, depends on the position of the introductory node. It seems to be a first, middle, last kind of representing different positions of the introductory. Now, remember, I told you that a very small fraction of birds don't produce any introductory. And what we did was to record in one of these birds. To our surprise, we found that there was activity before the start of song that resembled introductory. like activity. Here's the bird, it starts its song without any introductory note. But there's activity here, and there seems to be 3 peaks of activity. This is the average activity. Across many trials, what you see is 3 to 4 peaks of activity before the start of, suggesting that this bird also maybe, while it doesn't produce any vocalizations, it doesn't vocalize those introductory notes, there's actually something going on in the bird's brain, almost like the bird is rehearsing the start of his song. This sort of in that bird in 9 different locations that we recorded from in that bird's HPC. This was what the average activity looks like, and it compares very well with birds that produce introductory notes. Shown in blue here are birds that produce introductory notes. What you see is whether the bird produces introductory notes or not, the activity is very similar, suggesting that these again reinforcing our idea that introductory notes are themselves not used by the bird to calibrate its vocal organ, but for an experimenter, they're an indication of when song is going to start, or indication of the bird brain preparing to start. And for the bird itself, some of these introductory notes are vocalized, sometimes they're not vocalized, as in the case of the no introductory note. So essentially, I've shown you that in the neural part of this talk, I've shown you that this neural activity depends on the position of the introductory note, and there's introductory note-like activity in this part of the brain, even without introductory, suggesting that maybe the bird is mentally rehearsing something before starting a song. Okay, so this is as far as what happens during the introductory note. Okay, but remember I told you our approach was to say that this is a start of the movement itself, or the complex movement, the song, and we're going to step back in time and ask what are the neural events that happen before the song starts. What I've shown you is that these introductory notes that typically happen before the song, I've shown you a bit about what happens both in the behavior and in the brain. And so now, if we step back in time, what happens before the first introductory note? And again, that's involves recording from, we analyzed some of the data that we had with the recordings in HVC. What you see here are 2 more examples of neural activity along with the song. These are 2 different birds, 2 different neurons. What you see is this neuron again has some spontaneous activity when the bird is not singing. As soon as a little before the bird starts singing, this neuron stops its activity. And this is similar to what I showed you earlier. The neurons reduces its spontaneous activity. It shuts up even before the bird starts singing. Notice that there are no vocalizations here, yet this neuron has stopped. And there are other classes of neurons in that same part of the bird's brain which increase their activity before the start of first introductory. Again, the point I want to emphasize is that there is no vocalization here, yet these neurons are changing their activity. Across many neurons that we recorded in multiple birds, we see that there are some neurons shown in blue and black that increase their activity. This is 0 here on this x-axis, represents the time at which the first introductory note is produced. That's the first vocalization. Before this, there are no vocalizations. Yet, neural activity appears to start about half a second before the first introductory note. And the other neurons, like I showed you in the previous slide, that become quiet or silent about half a second before the start of first intro. And for those of you who are for details, those of you who are interested, these are inhibitory interneurons, the ones in black, and both the red and blue are a class of excitatory neurons in this part of the. So what we've shown essentially is that HVC activity changes about half a second before the first intro. To summarize my entire talk, I've told you that we're interested in how movements are initiated. We've shown that these introductory notes, or these short syllables that the bird produces before the start of his song, they appear to have consistent, they slowly change their properties, reach a consistent state, or before the last introductory note, what we call as a ready state, that seems to indicate that the bird's brain is ready to start his song. We've shown that this progress, this these introductory notes reflect something going on in the bird's brain as the bird brain gets ready to start song. We know that the number of these introductory notes is learned from a teacher or a father. And what I've also shown you is that neural activity in a particular nucleus of the bird brain, in a motor area of the bird brain, shows activity that's dependent on the position of the introductory note. This activity is present even when there are no vocalizations, there is no introductory notes, but there's introductory note-like activity in the bird brain. And even before this introductory note-like activity or the introductory notes themselves, we see neural activity beginning to change about half a second before the first vocalization. To summarize, our current model for song initiation is as follows. Uh, this is the cartoon of the bird's brain. There's some neurons that I've shown here in this area called HVC. These blue, each of these circles represents one neuron, and when it's blue, it's not active. When it's red, it's active. So suddenly something changes in the bird's brain. A couple of neurons become active. Then about half a second later, the bird there's again a change in the neurons that are active. This change in this particular area results in bird producing the first introductory node. Then again, a different set of neurons are active. Some of them may overlap with this, but some of them are different, like this one here. These neurons become active, and the bird produces its middle introductory node. Then a different set of neurons, again partially overlapping with this subset, becomes active. The bird produces last introductory node, and it goes on. And essentially, what we are doing in the next few years is to try and understand what determines all this. What is it that gets these neurons active in the first place, and how do these neurons decide which ones have to be active at which point? So, in other words, this sort of like this neural orchestra that's happening is this set first is active, these neurons are active next, and then this, and then this results in the song being. Who is the conductor for this? Which area of the brain is controlling this, or what are the mechanisms that control which neurons are active at which time and result in this as song being. What we're also interested in is we're looking at other songbirds. Because one of the things that you might ask is, are these introductory notes very specific to the zebra finch? So then it's a very zebra finch specific problem. What can we learn about this in general, be related to other songbirds? And to address that, what we're doing is to look at other songbirds. So songbirds, as I told you, are an order that consists of about 4,000 different species. All of them learn their songs from adult bird. That's why they're in the here. And here are two other birds, the English finch and the Java finch, that are related to the zebra. We recently got these birds also in the lab. These birds also begin their songs with introductory notes. By characterizing how their brain starts song, what we can hope to understand is general principles that are important for initiating song that are conserved across all of these different bird species. Neural mechanisms that are not specific to the zebra finch, but that are more general to all song. And to give you with a little more of how this is relevant to maybe our movements, what I'd like to show you is here's Rafael Nadal producing a different kind of movement, not singing, but he's producing a tennis serve. Again, this is a mixed movement, self-triggered because there isn't a person standing on the other side with a sign saying, now it's time to serve, now it's time to do this, etc. But instead, what Nadal is doing is he goes through this initial routine of bouncing the ball. In fact, he does a lot more that are not included in this. What you'll see here is he gradually bounces the ball multiple times, then he actually gets into his serve. And this is sort of, we can see parallels between the introductory notes that the bird produced and this ball bouncing that Nadal did. And this is prevalent in many of the sports that we perform. And one of the questions that we're also interested in understanding is, are these parallels just qualitative, or are there quantitative parallels between the introductory notes and these what are called pre-performance routines that are present before the start of a sports? To what I'd like to leave you with is to show you that, in fact, we've learned a lot about how simple movements are produced using controlled tasks like this in the case of the monkey and in the case of the rodent. And what I'd like to add is that our work suggests that the songbirds could be an excellent model system for understanding how complex movements, especially self-triggered movements, are. This is a cartoon that my son drew, showing that in fact the zebra finch may be a good model system for understanding a variety of different movements that are produced by us as well. I'd like to end with that to again thank all the people who've done the work. This is Divya and this is Shika. These are the people whose work I spoke about mainly. The other people whose work I've not had time to talk about. I'd like to thank ISER Pune for their support and a number of collaborators and a number of mentors who've been helpful throughout different stages of my career. I'd also like to thank my source of funding. And thank you all for your attention, and thanks once again to Shreya and the organizers of Brain Awareness Week for giving me this opportunity.